All right, so last lecture we were talking about HIV and the general concerns or problems with it and how it attaches to those CD4 or those CCR5 receptors and targets the CD4 cells, which primarily are the helper cells in your immune system. So it's a huge problem. Obviously, we have millions of people worldwide that are HIV positive, millions of people that have progressed to AIDS and died. So the goal is trying to figure out, how do we prevent these things? How do we stop these viral infections and help people live healthier and safer lives? So here's the general overview, and I'm not asking you guys to know the little details. You'll be expected to do that as you move on into upper level courses. But you look at the general process here. Attachment, get my pen here. So attachment to the cell because of the receptors. Entry into the cell, CD4 cells, these helper cells. So there's the viral genome, and it's an RNA-based virus, what we call a retrovirus. And that's what messed scientists up for years trying to figure this out. But what's going to happen here is the viral genome goes through a thing called reverse transcriptase. So RNA, in the normal world, think about the normal process. This is supposed to say DNA. DNA gets transcribed into RNA. RNA gets translated into proteins. Okay, hopefully that looks familiar or sounds familiar because it looks terrible. But with a retrovirus like HIV, it comes in as RNA. So then that RNA gets transcribed into DNA, then the DNA to this RNA, and then to the protein. So it has this extra step in there, which creates problems when we're trying to block it, when we're trying to understand it. It also increases the potential for mutations because now you got an extra step, extra possible location for mistakes and errors and things like that. So once reverse transcriptase has happened, the double-stranded DNA is blended or incorporated into the host DNA, and then it's holding on to it. And now it creates little viral particles, and they assemble. And then those viral particles get released, and those go out and attack other cells in the body and infect other cells. So when it comes to treatments, therapies, retroviral drugs, this is what we're looking at. Different ways of stopping it. So can we block entry? There's a drug that will stop that. Can we block entry over here? There's a drug that can stop that. Can we block replication? Can we block integration? Can we block the assembly, or as they're calling it here, maturation? So the key with this is each of these green X's represents a defense we can put in play against this virus. If you only have one defense, the offense soon figures a way around it, and then your defense is useless and you're shut down. So we have to have multiple drug treatments, multiple therapies to block it at all different levels, all different locations, etc., to try to make sure we can constantly shut it down. So if we can keep the HIV offense off balance with good changing defenses, we have a better chance of preventing this from becoming a major, well, even more significant of a problem than it is currently. So retroviruses, the thing I wanted to mention is that these guys use reverse transcriptase to create or to code for the viral genome. To code. All right, so again, it adds a big problem. HIV is a retrovirus that causes AIDS. The AIDS is what destroys your immune system, or when your immune system is destroyed, you have AIDS, and then you get sick from opportunistic infections. So hyperlink here, there's tons of YouTube videos out there John Hopkins has some great stuff. Howard Hughes Medical Institute has some great stuff. I encourage you guys, just take a look at some of the videos and some of the information out there. There's some really, really good information out there to give you a better overview of this. Some fact sheets, another hyperlink. Take a look at it if you want. I am not going to ask you guys to know these little details. This is for those who are interested in the topic to pursue it a little deeper. But what we're looking at is the statistics behind it. All right, sorry about that. I wanted to give you guys a more updated map here when pulled the 2017 data, just so you can see a little bit better update here on HIV diagnoses in the U.S. Couldn't get 20 data. This is 17 data, but it gives you an idea of what's going on around the U.S., where we're at in Illinois. Fortunately, we have, well, unfortunately, but fortunately, 9.9 per 100,000 people in the population are diagnosed with HIV. So roughly 10 people in Springfield have HIV. Compared to some other states, we're doing really good, but compared to some other states, we're also not doing so good. So, but the key, though, is looking at the different treatments. And there's 32 antiretroviral drugs out there on the market right now. And what they tend to do is target different aspects of the viral life cycle. So that's what we're seeing in that overview. One aspect is the viral entry. Can we block it at that point? Keep it from getting in? You're good. Can we block reverse transcriptase, or step 3 of the viral process? There's an antiretroviral drug for that. There's the integration inhibitor, and then there's also what's called the protease inhibitor as well. So the point being, when we're looking at HIV treatment, and what we're probably going to be looking at with lots of viral disease treatments, are combination therapy. The one for HIV is known as HAART, H-A-A-R-T, highly active antiretroviral therapy. When this is applied, and this is basically laying out as many defensive strategies as possible against the virus, AIDS death rates dropped by 75%. But when that treatment was stopped, viral levels have risen in the patient. So as long as we can keep that treatment therapy going, people can have an extended quality of life. If we can't provide that basic health care, then those people will see the virus levels rise, most likely progress to AIDS, and most likely pass away. At this point, vaccine development has been unsuccessful. It may be something in the future, but it's not something anybody should bank on, that, oh, well, we're just going to have a vaccine and everything will be fine. It takes a long time to get a vaccine that works, that is safe, that is effective. So those are things we really, really need to keep in mind. So, all right, so what I'm going to show you now is the big picture of other viral diseases. Just a table to use as a reference. There's 2 parts, there and there. We're not going to cover every one of them. We're going to talk about big picture, kind of highlight a few of them. I'm not asking you guys to know the little details. This is for reference. My biggest concern is, sorry about that, my biggest concern is getting a call in the middle of a lecture. Done with that. Let's keep talking about these viruses. My biggest concern is you recognizing which health issues are viral based versus which health issues are bacterial based, and so on. Because at this level, intro course here, I want you to be able to say, oh, that disease is a virus. We can't use an antibiotic on it. Or, oh, that infection is bacterial based. Let's get the right antibiotic. It's not to understand every little detail right now. You will be doing this later on as you guys progress in upper level courses. Those of you heading to med school, you're going to do it for a career. But for you guys at the intro level, let's just recognize what is viral based versus bacterial based. We'll talk about fungal-based diseases, protist-based diseases, prion-based diseases, and just recognizing what is the cause of the disease, and then generally, what do we do for it? Okay, so let's check out our first one here. And the first disease I want to mention is influenza, the flu. Unfortunately, very appropriate right now. The flu holds the record. Let's give it a little red star. Holds the record. Influenza holds the record for the most lethal virus in human history. So we go across human history, and influenza has killed more people than any other disease. Ebola, scary stuff. SARS, hantavirus, etc. COVID. Well, COVID's a type of influenza. All bad. But the influenza virus, the flu virus, is the worst. So flu viruses are enveloped inside, or they are an enveloped animal virus. So these are all animal-based viruses that can infect us because they have those recognition proteins that attach to our cell. Generally, when we look at influenza, there's 3 types, A, B, and C. They're classified based on the capsid proteins that they contain. Type A is the type that leads to epidemics, big problems, global problems. Types B and C are the types that lead to your general mild infections. So what we tend to see rolling around every fall are types B and type C. You get the runny nose, the headache, the fever, the body ache, the chills. Your immune system kicks it in a couple days. You're back on your feet. Hopefully, hope everybody doesn't get it, but you get it, you're back on your feet. The type A are the problematic ones. These are the nasties that really, really cause us the problems. This is what we're dealing with, an epidemic, or worse, a pandemic. So the mutations that occur on the viral particles, the capsid proteins, are what lead to problematic viruses. This is also the challenge with vaccination development. So here's an example, an original influenza virus. It's got a specific antigen right there. And we can develop a vaccine against that. So our immune system, this is supposed to be your immune cell, it can recognize that and say, hey, if I see that, I'm going to block it. I'm going to stop it. I'm going to attack it. I'm going to start the immune response and fight this off. But when these viruses undergo these antigenic shifts, notice the antigen is different. So now our immune system goes, I don't know what this is. It's like a whole new offense coming at your defense. And the defense doesn't know what to do with it. It has to learn, it has to figure it out, and that is why it's a problem. So when we look at the different pandemics around the world throughout human history, the Spanish flu of 1918, this has been inaccurately portrayed in news right now. The Spanish flu was in 1918. Killed millions and millions of people. It's estimated between 50 and 100 million people died of this flu. It's called the Spanish flu because of the publication of the flu and the information in Spain. It did not originate there. It's not from Spain, but they were printing information about the Spanish flu and educating people about it, about the flu in 1918, hence the name. The Asian flu in 57, the Hong Kong flu of 58, these get these general names because of where the outbreaks tend to happen. They're not caused by these regions. They just, unfortunately, suffer the initial outbreaks often from these viruses and these flus. Click the hyperlink, it was working. Hopefully it still works. It shows how flu attacks and how a virus invades your body. Pretty neat little video. What we'll talk about in the next lecture is the H5N1 bird flu, talk about some scenarios there, and then start working our way through some of these different viral diseases and talk about the problems associated with the viral diseases.